Okay, hi everyone. Welcome to today's webinar, Viral Surveillance Made Simple. Thank you so much to you guys for joining us. We're really glad you could be here. My name's Leanna, and I'm on the marketing team here at Plasmidsaurus. So before we get started, I just wanted to have a quick note on how today's session will be run. We'll save time at the end for Q&A, but you don't have to wait throughout the session. If you have any questions, feel free to type them into the Q&A box you see down below at any point during the webinar. So we'll be keeping an eye out through those, and Christy from our team will be helping run that session at the end. And one more thing to look forward to, all attendees today will receive a link for free Plasmidsaurus socks, which we'll share at the end of the webinar. So now let's kick things off. I'm pleased to introduce Sean Cordes, product manager here at Plasmidsaurus. Sean will start us off with a quick overview of Plasmidsaurus, and then we'll get to hear from Dr. Robert Potter of the Children's Hospital of Philadelphia, who will share how he uses long-read sequencing to trace norovirus transmission within the hospital. So Sean, over to you.
Thanks very much, Leanna. My name is Sean Cordes. I'm a product manager here at Plasmidsaurus. I wanted to talk just a little bit, give you an introduction to Plasmidsaurus. So like all of the best companies, Plasmidsaurus was born in a garage. Our CEO, Mark Budd, was working in cell therapy at the time and found that plasmid sequencing services available to him were not delivering what he needed with respect to quality and speed. And this insight led him to start his own sequencing services company, which over time evolved into the present day Plasmidsaurus. On this slide, you can see what it originally looked like in the garage. And fortunately, we have a much bigger setup now with multiple labs. And what's important for you to take away from this slide in particular is that critically, our founders are all scientists, and Plasmidsaurus as a company is fundamentally tied to filling a critical need for the scientists that we serve. We do this by constantly innovating to help scientists obtain higher quality research results faster and more efficiently. And we do this by constantly, improving both wet bench and bioinformatics analyses and various technologies to improve the scope, speed, and quality of sequencing. We're building a global network of labs with superior logistics to move your samples forward as quickly as possible. And that's really captured here. Today, Plasmidsaurus serves tens of thousands of scientists worldwide with more than 1,000 drop boxes and 10 local service labs. Our network of local labs means samples spend less time in transit. And for a majority of users, samples are delivered to a local lab and can be run overnight, getting you your results before you start experiments the next day. Contrary to our name, Plasmidsaurus is more than plasmids. And we sequence multiple different kinds of samples. Beyond plasmids, we sequence amplicons, which we'll be delving into a little bit more today, as well as both prokaryotic and eukaryotic genomes and microbial samples, as well as AAV. What remains across the portfolio is our philosophy and approach to sequencing with a real focus on returning high-quality results quickly. As I mentioned, today we'll focus a little bit on our amplicon sequence offerings. We have a couple of different offerings, which I'll describe more on the next slide. Turnaround time for these offerings is between 1 to 2 days, and pricing starts at $15 per sample. So the two offerings that we provide are diagrammed on the left side of the slide. The first one is called the linear PCR service. And this service is designed for sequencing clonal linear DNA, such as you might obtain from a patient viral isolate. And it's the service that Dr. Potter's been using for norovirus surveillance. It's basically a drop-in for Sanger sequencing with some real benefits over Sanger. The second service we offer is called the premium PCR service, and this is designed for more complex samples. So, for example, you might have a DNA sample with multiple amplicons. You might be doing, for example, whole genome sequencing of influenza, which has multiple segments. Or it might be DNA collected after CRISPR editing, which has induced genetic variation at the target locus. Both services have important advantages compared to traditional sequencing methods and services in that they're comprehensive, so you can sequence either whole viral genomes or amplicons anywhere from 100 base pairs up to 25 kb. They're incredibly fast, as we've discussed. They've been designed from the ground up to be really easy to work with. So everything from real simple ordering of your sample to requiring really minimal sample preparation prior to sending samples to Plasmidsaurus. So you don't need sequencing primers, there's no library prep, and you don't even need to purify your sample. You can send us your PCR product and we can sequence it. They're also incredibly flexible. So we have really minimal DNA input requirements, and we can work with multiplexed amplicons, as I described, for the premium PCR service. So with that, I'd like to transition the talk over to Dr. Potter. We're really excited that he's agreed to speak on our behalf today. Dr. Potter earned his PhD in molecular microbiology and microbial pathogenesis, followed by a clinical and public health microbiology fellowship, both at the Washington University School of Medicine in St. Louis. He then studied urovirulence and Proteus mirabilis before joining the Children's Hospital of Philadelphia, where he currently wears multiple hats. He's an assistant director of the Infectious Disease Diagnostics Laboratory at CHOP. He's the director of Outbreak Investigations for the Center for Microbial Medicine at CHOP. And he's an assistant professor in the Department of Pathology and Laboratory Medicine at the Perelman School of Medicine at the University of Pennsylvania. His clinical and research interests include the use of next-generation sequencing to improve patient safety and uncover novel insights into microbial evolution. So with that, over to you, Dr. Potter. Thanks very much.
All right. Thank you, Sean, for the introduction and sort of the brief background on Plasmidsaurus technology. The full title is Identifying and Interrupting Intrahospital Norovirus G2 Transmission. If I was to ever get invited back for a webinar next year, hopefully I can talk more about the interrupting part, because that's where we're currently working on, because fortunately, because of how successful we've been at sequencing things, the identifying part we've really got down now. So just for a bit of context, it's an unfortunate reality that the hospital is actually a very, very dangerous place to be, especially if you're a sick patient, which many people in the hospital are sick. So hospital-acquired infections, or HAIs, they exert a significant burden on patients and the healthcare infrastructure in the United States as a whole. This infographic has a variety of metrics that are used to quantify really how bad the HAI burden is. The two statistics that to me stand out the most is that about 100,000 people die preventable deaths due to HAIs, and about 5% of all admitted patients end up contracting an HAI upon admittance to the hospital. So it is a very big burden, and it's something that as clinical microbiologists, our job should really be to limit the amount of infection spreading in our patient environment. There are a lot of different ways that organisms can go from a colonized or infected patient into a susceptible patient. This can be through fomatic transmission on contaminated medical equipment, such as stethoscopes, which may go from room to room. It could be on the surfaces themselves in the room, such as door handles, sinks, toilets. And additionally, the other variable is that it could be the vector of both the healthcare workers and also any visitors, which at the Children's Hospital of Philadelphia, we're also, of course, concerned for caregivers as being the kind of vector for transmitting an organism from susceptible colonized patient into a susceptible patient. And so because microbial pathogens have evolved a plethora of ways to infect vulnerable hosts, we really need to be thinking about the best ways that we can understand these transmission patterns and then stop them and intervene once we identify them. But identifying and really determining if there an outbreak is occurring is also fairly difficult. It really depends a lot, in particular, on what the actual scenario is related to the disease, the patients, both temporal kind of arrangement and spatially where they are in the hospital. So, for instance, in this particular scenario, we have disease X, and if we say disease X is rare for this community, and say, for instance, patients A, B, C, and D, they're all on the same floor. And then temporally, we have patient A being positive day 0, patient B and C being positive day 1, and patient D being positive day 2. If the disease is really rare, it really does not take sequencing to say this is an outbreak. Something really must have occurred to result in these 4 patients being infected by this relatively rare disease. But unfortunately, what is far more common is that you get scenarios like this, where disease Y might be very common for this community. Patient A and B might be on the same floor, but it could be a different floor than C and D. And so now, although you have the same temporal spread of patient A on day 0, patient B and C on day 1, and patient D on day 2, because the disease is very common and because your patients are spread throughout the hospital, it's really less clear if this is an actual outbreak or if this is just kind of general biology occurring. So we then need to turn to whole genome sequencing as our tool for determining if something is the same organism or if it's different. Because with whole genome sequencing, once we have that resolution, if we can determine something's the same, then we can identify what is the actual transmission patterns. CHOP, the Children's Hospital of Philadelphia, has had a reoccurring issue with norovirus G2. This is an infographic showing testing data from last year in February 2024, showing that for the Northeast region in the United States, about 15% of all tests that look for norovirus were positive, which is a very high amount. This makes sense if you think that the Northeast region in the United States is generally the most industrialized portion of it, and norovirus spread via gastrointestinal tract, and so it makes sense, more density, more disease. Norovirus had a really big year in 2024 and then going into 2025, and so we are sort of expecting that that wave will continue to increase this upcoming norovirus season, which should start in a few weeks once really the weather cools down and fall hits. And norovirus really not just affects the patients, but it also affects the workforce. So this is an article that I would like to point out came from over 10 years ago in 2014, detailing how in this particular outbreak of norovirus at CHOP, although patients were also affected, about 20 healthcare workers were also affected. And so it's a big deal for the individuals who are providing care for our patients as well. Just some general biology on norovirus. It's a single-stranded positive sense RNA virus. It is non-enveloped. It's about a 7.5 kilobase pair RNA genome, and it really only encodes 3 particular genes. That blue gene is primarily the polymerase. There's some immune antagonism functions as well, RF3. Those form the capsid, and the capsid is this extremely tight protein coat that is what makes norovirus such a scourge for the hospital, because that capsid is so resistant to disinfection, it's so resistant to desiccation, it's able to survive for up to 3 weeks on a surface, and it's able to resist most of our cleaning solutions. And since it's 7.5 kilobase pairs, that's a bit longer, about 7 times longer than what the max of Sanger sequencing could really do. And previously, in order to sequence norovirus, you would really need to do some complicated primer pooling strategies where you can have a lot of these short amplicons that you're able to then sequence using Illumina technology. But those primer pools rely on a lot of different primers. And my opinion is that the more time you introduce opportunities for error, the more error that's going to occur, especially when you're operationalizing this type of testing within a clinical laboratory where we have about 65 full-time employees. And so it's much harder to kind of get increased granularity on when troubleshooting might be needed because something failed. So much simpler, we wanted to create an assay where we have just 2 primers, a very traditional PCR amplification. We have our forward primer on the far left end of that norovirus genome and our reverse primer on the far right genome. And then we just amplify it like a normal PCR reaction. And then we perform Oxford nanopore long-read sequencing in order to get complete sequences of that norovirus genome. So our goal is to use whole genome sequencing of norovirus G2 samples to aid infection prevention and control measures. The approach is to use full-length PCR amplification and Oxford nanopore technology sequencing. And then our hypothesis is that we'll identify spatially temporally linked outbreak clusters with 5 nucleotide SNP differences. And one way that all this is possible, coalesced when I joined CHOP in July 2024, was the creation of the Center for Microbial Medicine, which is a new center for emphasis at CHOP. The goal for the Center for Microbial Medicine is to use advantages in next generation sequencing and microbiome sciences to improve the lives of pediatric health, both through a clinical trials division, a basic science division, and also an applied division using sequencing to aid in improving clinical decision supports. And so this norovirus sequencing is an important early pillar in our attempts to use next generation sequencing to improve the livelihood of children at CHOP. So we have 2 different sample sets. We have our retrospective cohort and our prospective cohort. Retrospective samples were ones that we cleaned out of the freezer from 2019 to 2024. Prospective sampling occurred essentially 2 to 3 times a week, beginning December 2024 and proceeding throughout the 2025 norovirus season. And we're starting with norovirus G2 positive stool samples that are already positive on our clinical assay. We're then re-extracting RNA from these positive samples using a QIAGEN EZ2 Connect system. We are then performing an initial RNA into cDNA conversion using the QuantaBio QScript UltraFlex kit. Then we're taking that cDNA and we're performing full-length whole genome amplification using the replica HiFi tough mix. And then we're sending out that tough mix PCR product to Plasmidsaurus for the long read sequencing linear PCR order that Sean mentioned, with the optional add-on to do a PCR cleanup step. So the total cost to the laboratory is about $28 per reaction, and we get next day results. Our drop-off time is 4 p.m. Very often before I'm in the lab, by the next day at 9 a.m., I got an email from Plasmidsaurus saying my sequencing is ready to go. So yeah, really incredibly happy with that turnaround time. We have a few different analysis platforms that we're analyzing on the CHOP Linux system. Starting with least granular is just high-level genotyping. We move into a bit more granularity with maximum likelihood phylogenetic tree. And then most granular is to look at the clinical metadata and to identify temporal spatial SNP analysis. In the interest of time, can't talk about everything. So we're just going to focus on the temporal spatial SNP analysis. But first, really the metric we wanted to see is, did it work and how often did it work? And so the way that we kind of measure that is how often samples, when we'd send off to Plasmidsaurus, come back as assembled or if they had failed sequencing. And then we take those two cohorts and we can segregate them based off the CT value to really see sort of the CT value as a proxy for viral load. So a low CT value means high viral load. And so it really does not take a PhD to see that there's a far, far lower average CT value for samples that assembled compared to samples that failed sequencing. It really seems to be that cutoff of around 30 CT value. Our odds of a sample failing sequencing occurs much higher than the odds of it assembling. But since we did have some sporadic successes, what this means to us going forward is that we will go ahead and try sequencing on all positive norovirus G2 samples regardless of CT value. But if it doesn't work the first time, it's likely it may not work the second time. And our clinicians are now understanding that. We didn't think of this idea. It came from this really great paper from Public Health Canada, our colleagues up north. We were really happy that we were essentially able to take that protocol where they tested 57 samples and had 39% work using in vitro supplies to then improve it to 84% success rate using QuantaBio supplies. And one of the major differences is that the long PCR that we're using ends up cutting the PCR reaction time from 6 hours to 1 hour, meaning that our sort of vision of a same-day turnaround time from a patient positive, next day we get the result from Plasmidsaurus, really is a reality because we can do all the processing in-house the same day that that patient is positive. So norovirus is a very complicated genotyping scheme that we won't get super in-depth, but really it relies on classification from the genotype, from the polymerase, and also from the capsid. And then that gives you sort of an alphabet soup of Roman numerals, locations, and P numbers that then kind of fill out into these genotype information where we have, for instance, here it's G2.4 Sydney P16 strain. This is the most common genotype around the world, so it makes sense that it's the most common genotype we see at CHOP. In total, we identified 30 different genotypes, but really this big 8 are the most common that we see. And then moving into SNP counting, what we're doing is really not that complicated. We're basically just taking 2 genomes, we're aligning them, and then we're just counting the number of SNPs or single nucleotide polymorphisms. So shown here, example, this is 2. But we're not just doing that on 2 genomes, we're doing that on about 500 or so genomes, and 557 genomes, which ends up giving you about 310,000 pairwise interactions. And when you distill those 310,000 pairwise interactions into a histogram, you get this plot, where we can see that there's sort of a varying levels of norovirus SNP distance. And fortunately for outbreak investigation, we really only care about SNPs that are down here. We only really care about the smallest bucket of most closely related genomes. So let's zoom in onto those norovirus samples. So this is a very complicated figure. I really like it, but I've stared at it a lot more than you all have before. So just to walk everybody through it. So this figure is a histogram or a cluster map that shows each of those individual dots is a particular norovirus genome. R's are our retrospective cohort from 2019 to 2024. P's are our prospective cohort of samples that we've just been sequencing. They're colored based off of the location. So black is inpatient, meaning they're in the hospital. Dark gray is emergency department. And then light gray is outpatient. Blue, there's a few samples we didn't get that information. And then the lines connecting the dots are the SNP distance. So red is 0 to 1 SNP, blue is 2 to 3, gray is 4 to 5 SNPs. And what's really remarkable is that about 26.2%, so just over a quarter of our cohort, falls into this really, really highly related clusters of super related genomes. So it's indicating that there's a lot of transmission that's occurring inside of the hospital environment. In total, we identified 7 clusters containing 5 or more genomes, 2 quartets, 8 triplets, and 31 pairs. And I'm all very new at this. And so what was really remarkable to me was that learning from CHOP's infection prevention control, that in order for something to be on their radar, it needs to have at least 3 incidences of infection occurring. And so we're essentially identifying 31 pairs of what we're calling cryptic transmission, where these kids are transmitting norovirus to one another. And it's just, we're very fortunate that this did not lead in this particular scenario to extended outbreaks. But there's really no reason to think that any of these 2 pairwise interactions can't suddenly blossom into a large cluster of multiply infected kids. So this overall shows, and then we kind of grouped them based off of the genotypes. It's really not surprising that that Sydney strain we mentioned earlier, it's the most common genotype we see at CHOP, and so it makes sense it's going to be the most common genotype causing these really highly related outbreak clusters. So this shows kind of overall relatedness, but it doesn't show temporal or spatial information. And so in order to get at that, we took a rules-based approach where we merged the clinical informatic metadata with all of our genomic sequencing data. And then in doing so, we learned that of those 50 clusters I showed, 5 of them are actually fake. It's the same patient that we then tested multiple times because we just tested everything we got. And then after the fact, looked to see if they're the same patient. So really, it's actually 45 true clusters. And then we looked to see if they were kind of considered likely, possible, or not likely transmission event. And so this graphic represents each of those different clusters. The dots are colored based off of the genotype. The graphic on the left side depicts our transmission that we'll kind of walk through shortly. And then the location shows that inpatient, ED, outpatient as different figures. And really the temporal information is helpful because we can see, for instance, that this cluster down here in the bottom, that's really not likely to be a transmission event. So for instance, this is basically indicating that the first index cases that are from an unknown area are around late 2019. But then the second cluster is from late 2021. And so that's really just kind of random chance of when you're sequencing highly related clonal populations, you're going to stochastically see some people that are related to one another. But because we know norovirus really only has a 3-week infectious window, this is not likely a transmission event. And so of those 45, about 19 out of 45 either had an outpatient sample within that cluster or were occurring greater than 3 weeks apart. So we said these are not likely transmission events. 12 out of 45 of those are what we're considering possible transmission events. And this is because they occurred within 3 weeks of each other. So you can see all those dots now, in contrast to that earlier figure, all those dots now are much, much tighter temporally. So possible transmission events occurred between 2 different inpatient floors, maybe say floor 3 and floor 5, and also the emergency department, which is a separate portion of the hospital, and any of the inpatient floors. And then finally, we have 14 out of 45 events that we're calling likely transmission events. These are the ones that are occurring within 3 weeks of one another, and they're also occurring within the same floors. So like 3rd floor to 3rd floor. These were the scenarios that IP&C were able to identify likely prior to sequencing, really to a high degree, but now we're able to provide a much greater granularity. And again, we're also able to identify now cryptic pairwise transmission events. We don't need to have that threshold of 3 kids being infected before triggering an investigation. And so we really wanted to close by just walking through this details of what, according to that rules-based classification, is a possible norovirus transmission event. But really, kind of when you look at the evidence, this is sort of exactly what we wanted to capture by sequencing. So we starting with patient A. Patient A is presenting to the CHOP ED. It's winter 2024. They're relatively healthy. They have symptoms of gastroenteritis. They order the stool viral PCR testing, and then they're discharged. Patient B is very medically complicated. Ironically, patient B is an infectious disease-related issue. They have a JG tube site that is infected. We ended up culturing Pseudomonas aeruginosa. Because they're medically complicated, they get admitted to the hospital for IV antibiotics. However, patient B just sat in the ED in that same room that patient A left. The next day, we result patient A's norovirus PCR as being positive, and then patient B, who's been in the hospital for 3 days, ends up developing gastroenteritis on their fourth day of admission. The clinicians order norovirus PCR. It's positive that same day. We do our whole genome processing and send out to Plasmidsaurus. Day 5, we determine that there's no SNPs, 0 mutations between patient A sample on day 0 and then patient B sample on day 3. So we email our colleagues in infection prevention and control. You can see this is the exact email thread. 11:49, she tells me she's going to look into it. 11:52, 3 minutes later, she emails back, already uncovering the investigation that patient B and patient A likely overlapped spatially in that particular room in the ED. And so otherwise, before sequencing, this never would have been noticed because it's only 2 cases. It's not that threshold of 3, and it's occurring in the ED in the hospital. So it would have been far more difficult to link these 2 cases to each other. And so now this teaches us that there is a particular room cleaning protocol, but there's a separate room cleaning protocol for kids who are positive for norovirus. And so to us, it indicates that maybe just because in the winter background norovirus is so high, we need to do the really intensive and expensive cleaning in order to kind of thwart any potential of norovirus spreading in the hospital. So in conclusion, hopefully I've shown you that whole genome sequencing of norovirus, it's cost effective, it's relatively quick. We have an 84% success rate. That Sydney strain is the most common genotype we see at CHOP, with other genotypes varying annually. And the SNP distance is really good at showing high clusters of highly related genomes within the hospital. Currently, we are training the clinical laboratory staff on this protocol. Previously, we've been doing it by myself and 3 of our R&D technicians. Now we're training the staff to do it every single day. We're continuing to perform this on all positive norovirus G2 samples. We're working on some additional assays for other viruses we want to sequence and for bacterial and fungal identification. And then we are uploading all of the Plasmidsaurus read data into NCBI so that it can get funneled into the CDC National Surveillance Network through CaliciNet. And really, why Plasmidsaurus? It's number one, comes down to the people. I really like what Sean mentioned as being a company founded by scientists for scientists. I emailed somebody this morning for help and they responded within like 10 minutes. Everybody I've interacted with is super intelligent. They're always very helpful, really, for anything related to like long-term project design, immediate understanding of maybe why did my particular sequencing run maybe fail, and then really great troubleshooting on any sort of technical discrepancies. It's fast and it's also reliable. It's one thing to be fast, but if it's not reliable, that's harder for me to then turn to my clinician colleagues to tell them why their test may be slower. It's really very consistently within that 12 to 16 hour drop off for the amplicon sequencing. And the continuous scientific development, I think, is really fascinating. Every time I go to the website, it seems like there's either a new assay. For instance, I'm really excited about those microbiome assays. And the continued upgrade of enhancing the site to be user-friendly for ordering and for analysis, I think is really nice. And really, I love all the Dino art gallery. I've submitted a few stuff. It hasn't gotten put on the Twitter, but I need to improve my artistic skills or hire some artists in the laboratory. But I like looking what other people do. It's very fun to see scientists flex their creative sides. And I would not be able to share all this exciting data without a fantastic team at CHOP who's supporting all this efforts. So I especially like to shout out my 2 other directors, Rebecca Harris, KP Smith, our laboratory leadership, including Carrie Thompson, Jackie Bazzoli, Jess Llewellyn, and then our research and development staff, Jeff Fink, Rose Patrick, Mike Elkin, Adriana Caruso, and Ana Luisa Jernigan, our colleagues in infectious disease, my colleagues in the Center for Microbial Medicine, and then collaborator at Children's National Hospital. And if there are any follow-up questions, if we have time, I'd be happy to answer them in the chat, or feel free to contact me via email. And thank you all for your attendance.
Thanks very much, Dr. Potter. So I think, Christy, I think we have time for some Q&A. Are there any questions?
Yes, so our first question is, do you have plans to extend this method to other viruses or even bacteria like CROs or other AMR organisms?
Yeah, we are very interested. So right after we got the norovirus sequencing down in December 2024, we then had this big push for avian influenza because that's also around the time when avian influenza was shown to jump into cattle. And so then there's additional concern if it can go into cattle, can it go into humans? And so the CDC and the state of Pennsylvania released guidance to hospitals saying, please type your influenza assays, please type all positive influenza samples. While we're working on a qPCR, to me, qPCR is scarier to me than whole genome sequencing. I love sequencing. And so it was far easier for us to implement a sequencing assay. And so we very quickly devised a intake form where if a kid was positive for influenza, the ID team would go into that kid's room. They would then talk to the family, see if there's any particular risk factors for avian influenza. We had, I think, at least 5, including some kids who like their best friends were chickens. And so if we thought if there was going to be pediatric avian influenza, it would be like this kid. And so we would use those for the um, additional PCR sequencing assay where we get the demultiplex reads and then construct influenza genomes to thankfully determine that all of those were either H3N2 or H1N1, but no H5s. And then we were additionally working on some ITS and some 16S sequencing for rapid bacterial and mold identifications.
Thank you. So our next question is, can you elaborate on why you think a single primer pair assay is superior to primer pool strategies?
Yes, superior, I think, would depend, at least for me, the assay needs to work, and it needs to work without me doing it, and it needs to work with somebody who just graduated from an MLS program and now is starting work in the laboratory. And anytime you can make something more complicated, there's going to be more opportunities for a technical failure, either in like the people side or like a mutation in one of the viral regions that makes that particular primer now no longer bind to it. And so when devising this assay, we really wanted something that we can implement confidently in the clinical laboratory and really kind of lower our possibility of there being an error. And so I think it makes sense that if you, instead of having 15 different primer pairs with 30 total primers, if you just have 2 primers, a single primer pair, it's much, much simpler to orchestrate.
We got another question. The clustering's based on, so I think for your SNP-based clustering, the question is, are there any regions that are more susceptible to exhibit such SNPs?
Yes, fantastic question. And there is. So in the very beginning of the talk, I showed that kind of general outline of a norovirus genome. We saw gene 1, gene 2, gene 3. There was that poly A region. And so most of the variation that we ended up seeing is in that poly A tail. And then it's really fascinating because it's both the kind of the biology of the virus, where it likes to have sort of varying numbers of that poly A tail sequence. And then also us as sort of, I like to say that really SNP thresholds are sort of where you put the philosophy and the doctor of philosophy part, because it really seems like it's very organism specific. We started off based off of me calculating what you're sort of expected to see based off the evolutionary rate of norovirus. But my calculations ended up being about like 30 SNPs, and we applied that. You get way, way too much clustering. We distilled down to 10 SNPs, but really it seems like it's more often 0 to 5. And so another alternative analysis that we do is sometimes we'll just clip off the leading and trailing 100 base pair SNP regions, so that then we're only analyzing the interior portions of the genome. You get rid of that poly A tail. We ended up still keeping the poly A analysis because generally that analysis still tracks, but oftentimes that's the particular region where you see the variation in. Great question.
Do we have time for one more question?
I think we can do, yeah, one more.
Okay. Have you considered to combine short read sequencing and long read sequencing in order to get more accurate results in regards to SNP detection?
Yeah, that was really the premise of that Public Health Canada paper. And so really, I think that this chemistry in Oxford Nanopore technology has improved considerably. It's crazy to think about where it started. I started my PhD 10 years ago, like this month. And I remember at the time we called the MinION a random letter generator. And so to now see how high fidelity the chemistry have is pretty remarkable. I'm sure that if you were to do an exact comparison, you might find some differences with Illumina sequencing, but that's also why for this particular project, I like to say it's a transmission study. It's not an evolution study. And so if there is sort of a systematic error that is only found in ONT compared to Illumina, I'm really only cared about the other chopped genomes, less so about norovirus evolution as a whole. But really, I think that now the chemistry is just accurate enough that that's not as big of a deal. There are some stuff where we are doing Illumina sequencing on, but that is sort of more of a feature of why we particularly want short reads compared to why we might want the Illumina chemistry compared to the nanopore chemistry. I think now it's really, really good. And it's also tough to say because for something like an RNA, for norovirus, it's an error-prone RNA virus. And so you kind of already expect a very high background level of mutation. And so then it is, again, very philosophical of knowing how is this, do we know this is a technical mutation and not just a biological one?
Okay. Thank you so much, Dr. Potter.